Hi there, welcome to session 3 on the cardiovascular system. Can I just check that you can hear me okay, please? Can you just let me know in the chat if you can hear me? Perfect. Okay. So last session we finished off we were about to have a look at the electrical conduction of the heart. So this is a little bit of a tricky topic. Again, loads of new words. There always is in biology because we have to label everything. What I would suggest to deal with all the new words, because they're usually labels, they're usually names of structures, I would learn them. Just sit down and learn each diagram and learn it off by heart like you would when you used to have spelling tests in primary school. It's the easiest way because you commit it to memory then, and it's there. And when someone starts talking about some random structure, like I don't know, the um, endoplasmic reticulum, you're like, oh yeah, okay, I know what that is. And it's not a sort of abstract thing in your head that you have to remember to look up later. You've got it there committed to memory. So I would try and remember all these names of structures. And I know it's a lot at the minute because this is quite intense. The course content is quite broad and there's a lot to do. So unfortunately, we're going to go on another topic with loads of new words and labels. And that's just after doing the heart beating, where we had all the new words for the vena cava and the pulmonary artery and the aorta and things like atria and ventricles. So another load come in today, I'm afraid. So apologies in advance for that. But it is an intense course because if you're thinking about it, think about it, you're trying to get up to the level ish of somebody who's been doing A levels for 2 years in about a year. So it is quite intense. So I'm just pop this into slideshow mode. And let's go from this slide because we're halfway through this slide set. So we're looking at the electrical conduction in the heart. And before we go on to that, I need to talk to you a little bit about ions. So this is chemistry. And the reason I'm talking to you about this is because I don't think you've started chemistry yet, have you? Can you let me know if you've done some chemistry sessions yet with because I don't think you have. Let's have a look what was in the chat there. No, not yet. Okay, so I need to do this really as a little bit of background. You might remember it from school. You would have learnt it probably around year 9, year 8 if you were in the UK system. So it's not, particularly advanced or anything, but you do need to know what we're talking about because we're talking about charges today. So that's an atom. Atoms are what make up everything. They are what the periodic table has got in it. So we're talking, I've got one in front of me because I teach science all the time. So I've always got one in front of me. So we've got hydrogen, lithium, sodium, potassium. What else can I see this end? Magnesium, calcium, all of those things. These are all elements. And the elements can be broken down into the smallest part of an element is an atom. So that's what we're talking about. The elements in this smallest unit is an atom. Now, atoms themselves can be broken down. They can be broken down into protons, neutrons, and electrons. But obviously, those are protons, neutrons, and electrons. But that could be an atom of magnesium. It could be an atom of sodium. It could be an atom of calcium. Yeah. So you see what I'm saying? As soon as you break that down further, you're talking about subatomic particles, but you could still be with atoms, you could still be talking about an element, but it's just its smallest form. So protons, neutrons, and electrons make up this atom, or any atom for that matter. So the nucleus is the bit in the middle, and that contains subatomic particles called protons and neutrons. And we have electrons that move around the outside, and they move around in, well, I won't go into too much detail because your chemistry teacher can deal with this, but they move around in what at GCSE we might call orbits or shells. When you learn chemistry, he'll tell you it's energy levels, or she'll tell you, I think it's a guy. So the electrons orbit around with different energy levels, essentially. The protons and neutrons are found in the middle, in the nucleus. You can see I've got little positive and negative signs on some of those particles. So the electrons are negative and the protons are positive. The neutrons are neutral, so neutrons neutral, helps you to remember it. If I was to look up a element on the periodic table, it would look something like this. It would have a big number and a small number. So they're not necessarily the big number at the top and the small number at the bottom. Just to make things hard, they can put these numbers in either place, so they can switch them around, it doesn't matter. But on this particular element, which is carbon, we've got the big number at the top and the small number at the bottom. Now, this small number tells me how many protons this element has got in its nucleus, and that is also equal to the number of electrons it's got going around the outside of the nucleus. This big number is the number of protons plus the number of neutrons. So I know that carbon has got 12 protons and neutrons, that's the total. And because I know from here that it's got 6 protons, I can work out that it's also got 6 neutrons, because 12 minus 6 is 6. So it says down here, that's what this number tells me, that there's 6 protons. So I can work out the number of neutrons really easy. And one other thing this number tells me is there are 6 electrons going around the outside of carbon atoms, because the number of protons is always equal to the number of electrons, unless some changes have been made. But your chemistry teacher can deal with that. So carbon has 6 protons. That's what that number tells me. It has 6 neutrons and it has 6 electrons, because that is equal to that number. I've worked out that it had 6 neutrons because this big number here is the number of protons plus neutrons. Okay, so hopefully that wasn't too taxing. But the point I'm getting to is this point here. Atoms have no overall charge. They've got charged particles in them. We can see the charges here. So I think this one is actually carbon. Yeah, I've drawn a carbon atom. So this has got 6 positive charges in the nucleus and 6 negative charges. Has it got 6? I can't actually see. No, it's not carbon, but it's got positive charges in the nucleus and it's got negative charges around the outside, and they'll be equal in number. So for this particular atom, carbon, 6 protons, it tells me that they're positive, and I therefore know that it has 6 electrons, they're negative. So if I add those charges together, -6 plus +6 equals 0. So there's no overall charge on that atom. So hopefully that makes sense so far. Are we all good with that? Can you let me know in the chat? Does it make sense? Overall, no charge on the atom? Okay, good. So the number of positive atoms cancels out the number of negative atoms, so there's no overall charge. So I've drawn them on the atom here. So we've got 6 protons in blue, 6 neutrons in purpley pink. I've worked that out, remember, because that's the number of protons plus neutrons. And we've got the 6 electrons orbiting the nucleus of the atom. They don't float off because they're negatively charged. So they're going to be attracted to the positively charged nucleus there. Now, if you look at my electrons, you can see that I've got 2 electrons orbiting in the first shell, and I've got 4 of my 6 electrons orbiting in the second shell. That's because when electrons are orbiting the nucleus of an atom, they always go in a arrangement of 2 electrons in the first shell, 8 electrons in the second shell, and 8 electrons in the third shell. So, sort of, your chemistry teacher will deal with that with you though. So they go in this 2-8-8 pattern as far as we're concerned for biology. That's as much detail as we need to know in biology. So 2-8-8. So this atom has got 2 electrons in its first shell and it's got 4 electrons in its second shell because it's only got 6 altogether. So it starts filling up from the inner shell. So it fills that up. It only takes 2. And then we put all the rest in there. We could put 8 in there if we had 8, but we've only got 4 left. So that's where they go. Now, that's what we call the electronic configuration of carbon, 2, 4. So we've got 2 electrons in the first shell, 4 in the second shell. We're going to have a look at ions now, which is how we end up with a charged atom, because we're going to be looking at charged atoms or ions today. So sometimes atoms can either gain or lose electrons, and they do this to get what we call stable electronic configurations, and that means they do it to get a full outer shell. So if we go back to our carbon, that outer shell is not full. It's got 4 electrons. Remember I said it could have 2, 8. You know, that's the number it could have to be full. So it's only got 4. It could fit another 4 electrons in there to make it full. Now, some atoms that have only got 1, for example, electron in their outer shell, are really reactive because it's quite easy for them to lose that 1 electron and they become charged quite easily. And 2 of those electrons that have, 2 of those atoms, sorry, 2 of those elements that have only got 1 electron in their outer shell are sodium and potassium. And they're really important in biology. So we're going to have a look at how sodium and potassium, well, just looking at sodium, but potassium, it's pretty much the same thing, how they form ions. So this is sodium. Sodium has 2 electrons in its first shell, and then its second shell is full as well because it's got 8. Remember I said it goes 2-8-8. But if you look at its outer shell, it hasn't got 8 electrons, it's got 1. So it goes, instead of going 2-8-8, which would be 3 full shells, it goes 2-8-1 electron. So in order to get a stable electron configuration, in other words, full outer shell, the easiest thing that could happen to sodium is it could lose this 1 electron. I mean, it could gain another 7, but that's not very easy. It's much easier to just get rid of this one. So if we get rid of that, this sodium atom will have an electronic configuration of 2-8 instead of 2-8-1, which is what it should have. So 2-8-1 is the electronic configuration of the sodium atom. But if it loses an electron, it ends up with an electronic configuration of 2-8. And we write that as 2-8 plus, which sounds weird because we've lost an electron. So why would we write it as 2-8 plus? We haven't gained anything, we've lost something. But the reason we write it as plus is now it has got an overall positive charge. I'm going to skip this one and see if I can go through to, yeah, no, I wanted to go through to this page. So how has it got an overall positive charge? Well, here is the symbol for sodium. Notice it's Na is sodium as well. So that's a bit of a tricky one to remember. So remember this number here tells me how many protons this element sodium has, and it has 11. And this number here tells me how many protons plus neutrons. So I can work out how many neutrons it's got. So I can do 23 minus the number of protons, which is 11, and however many, I get as my answer, which is 12. That's how many neutrons it's got. So it's got 12 neutrons. It's got 11 protons because it says so there. And it also has 11 electrons because they're always equal. So it's got 11 protons, 11 electrons, and I've calculated it's got 12 neutrons. So these 11 protons are all positive. So it's got 11 positive charges, and the 11 electrons are all negative. So it's got 11 negative charges. So if I do negative 11 plus 11, the overall charge of a sodium atom is 0. However, if you remember back to our previous slide, our sodium atom lost its 1 electron in the outer shell. So it was 281, and really it wants to gain stable configuration and become 28 or 288, but it's much easier to become 28 than to gain another 7. So it's become 28, and we're writing a little plus there, and I'm trying to explain why. And the reason for that is sodium has 11 protons. The number of these hasn't changed. You can't change that. It wouldn't be sodium anymore if we changed that number. It would become another element. So it's got 11 protons, 11 positively charged protons, and we've just lost 1 negatively charged electron. So we've gone from the 11 electrons that we started with to 10 electrons. So now we've got negative 10 plus 11 gives us an overall positive charge. So sodium as an ion, that means a charged particle, has a +1 charge. So we don't write +1, we just like write a plus. But if it had an overall charge of +2, we would write +2 or +3. But with 1, we don't write the ones in. So it's got 10 electrons, which are negatively charged. You can count them here. Look, 2 in the first shell, 8 in the second. And it's lost that extra one from here. And it's got 11 protons. So 10 negative charged particles and 11 positive charged particles. So an overall charge of +1. So sodium atoms can lose an electron to become a positively charged sodium ion. So apologies for the impromptu chemistry session, but we need some chemistry to do some biology. We call positive ions cations, by the way, which is why I've got a positive cat there to help remember that. So I'm going to skip over sulfur because it's not that important for what we're going to do today. We're going to have a look at the electrical conduction of the heart. So before we go into the conduction, I just want to remind you of the flow of blood through the heart from last session. So we're just going to quickly watch this. So apologies in advance for any ads here. Have a look what happens to the heart now when it contracts. That contraction starts from the bottom of the heart. It squeezes upwards. Okay, I'll stop it there because I don't need to go through that bit of systemic circulation. Right, so when the heart contracts, we have these phases. We have systole when it's contracting, that's the bit that pumps the blood. And when it relaxes, we have diastole, which I've heard some Americans pronounce as diastole. So diastole is how I heard it pronounced usually in the UK. So diastole and systole. So diastole, the heart chambers expand to receive the blood. Systole, they contract, or the way I remember it is systole squeeze. So that's when we get the contraction. And we've had a look at this circulation over the last couple of sessions. So the heart is circulating the blood up around to the lungs for oxygenation. It comes back to the heart. Once it's picked up oxygen, goes off around the tissues in the body. So when the heart is squeezing and filling, both the atria and the ventricles are undergoing systole and diastole at the same time. So you haven't got one side squeezing and the other one filling and, you know, this sort of thing going on. They both open up to fill. Then there's a big squeeze from the bottom. Nothing's happening independently, even though there's two separate circuits, one going to the lungs and one going around the body. The heart organ squeeze is in a coordinated way. So it's essential that these components are carefully regulated and coordinated to ensure that the blood is pumped efficiently around the body. So a complete set of pumping and filling. So diastole, the heart fills with blood. Systole, it pumps. That complete filling and squeezing out the blood is called the cardiac cycle. So here comes the reason we were looking at charged ions. Now, this is a neuron, and we're not looking at neurons today, but I couldn't actually find a image like this with a muscle cell, which is what we're looking at. We're looking at the heart muscle. This shows a different cell from a different type of tissue, but nerves are able to conduct impulses because of depolarization across the membrane. And so are these specialized heart tissues. So all animal cells have a membrane. We've talked about that in one of the first sessions that we did. There's a cell membrane. It controls what enters and exits the cell. And one of the things that can enter and exit the cell are charged particles, like that sodium we were looking at a minute ago with the chemistry stuff. So that sodium ion is what we call a charged particle. So they can leave and enter the cell across the cell membrane. And the fact that they are charged means that there is a membrane or cross-membrane, transmembrane potential, or a membrane voltage due to there being a difference in electrical potential between the interior and the exterior of the cell. So what that means is if the outside is positive and the inside is negative, there's an electrical potential. We've kind of experienced this through static electricity. You know, when you get a zap, when you, I don't know, you rub your feet on some like nylon-y carpet in a hotel or a school corridor or something, and you've got plasticky soles on your trainers, you can get an electric shock, or you get them off cars because they've been driving past particles in the air. You can get electric shocks of car doors sometimes because static electricity builds up. That's the kind of thing we're looking at. We're looking at charged particles. So the interior potential with respect to the exterior of a cell is usually negative, and it usually has a charge of about minus 40 millivolts and 90 millivolts, minus 90 millivolts. So that's really small. If you think about your electricity that runs through your house, that has a voltage of about 230 volts. So we're looking at minus 40 millivolts or 90 millivolts compared to 230 volts. So it is really, really small across the cell, but it's enough to send an electrical signal around our bodies. So yeah, that's basically what it says. The electrical excitable cells, such as neurons, which we can see here, and the muscle cells, including the cardiac muscle that we're talking about today, is how signals are transmitted across them. And we call this impulse, this potential, this ability to change from positive to negative, an action potential. So that's the name of this charge difference across the membrane, the action potential. So what this action potential is essentially, it's a brief depolarization. So that's what you can see on the little moving image there. It's a brief change in polarity or charge. So it briefly switches from positive to negative across the neuron in this instance, but we're talking about muscle cells today. So it's briefly changing, and that change moves along the length of the cell, length of the neuron on the image. So it's a brief change or depolarization of the potential across the cell membrane of the heart cells that we're looking at today, which causes the movement of charged atoms or ions, like that sodium ion we looked at a moment ago, between the inside and the outside. And how do we move these charged atoms or ions across? Well, they move through something called ion channels, which are special proteins in the cell membrane. And you can see some here. So on this image, A is a sodium ion. So that's sodium that has lost its outside electron. So it's got an electron configuration, not 281, but 28. So it's positively charged. And you can see there's loads of sodium ions on this side of the cell membrane. This is the cell membrane, and it's got loads of proteins inside it here. So we've got loads of sodium positively charged. So the outside of this cell is positive compared to the inside, which is more negative. The inside has actually got some potassium ions here. This is what B is. So A, I'm getting it off the key here. Look, A is sodium, B, which is this little triangle, is potassium. So potassium is positively charged as well, but the inside is more negative because there's loads more sodium than there is potassium. We've got all these channels. And what these channels do is they take sodium across. So you get this. They're taking positive ions across, as you can see here on this neuron. So the positive ions are moving across the cell membrane into the cell. So that's what's happening here. We're taking these positively charged sodium ions into the cell across this cell membrane. And you can see the inside of the cell becomes positive because we've got all these positive potassiums, which were there anyway, but now we've got all these positive sodiums as well. So the outside becomes negative compared to all of these positively charged ions that are inside the cell. So have a look at this. You can see the wave of depolarization becomes positive on the inside compared to the outside once those particles, those ions move across. And that's what we've got here. This is the positive bit of the depolarization. Then we use this pump. It's actually a sodium potassium pump, the red one, E here, sodium potassium pump. And what this does is it pumps sodium across the membrane. Also takes a little bit of potassium with it, and the outside becomes positive again. Then we have a special potassium channel, which gets the potassium where it needs to be, so we can start the process again. So that's how it kind of works. You don't really need to remember that, but it's just a sort of explanation of how this charge can change across the membrane. So the cardiac action potential differs from this one that we just had a look at. and it's got nothing to do with nervous activity. That's really important, because if that was the case, if people had a spinal injury, for example, their heart would stop. So there's no nervous stimulation of the heart. It arises instead from a group of specialized cells, and these cells automatically produce their own action potential. So there's no nerve signal. And these specialized cells are found in the right atrium. You need to know where that is, where these cells are, and they're called the sinoatrial node, or the SA node. Let me just check the chat. Are the sodium and potassium recycled? Yeah, they're recycled. Yeah. But yeah, we can gain them from nutrition. Yeah, but they're recycled and reused. Yeah. So sinoatrial node is where this starts. It's in the right atrium. I'm going to have a look at a little video of this now. One second, sorry, I just got a question coming in. Okay. Right, I think the video is not sharing the audio for some reason, so I'll talk through. That's fine. Right, so what we've got there is the sinoatrial node, which was up there. That's the bit that I was telling you about. This is a bit of specialized cells. This is where the heartbeat is initiated. This here is the AV node. That stands for atrioventricular. Remember, this bit's called the atrium, and that's called the ventricle. So it just means it's going between the atrium and ventricle, atrioventricular. The reason that is there, look, you can see the impulse spreads across the atrium. That's fine. But then we need to get this down to the bottom of the ventricle. We can't just let it spread over the ventricle, or otherwise the ventricle will be squeezing from the top down. We can't let that happen. So it goes down this fiber from the AV node, the atrial ventricular node, and this fiber is called the bundle of His. And then it goes up to these fibers at the bottom called the Purkinje fibers. And unfortunately, they've all got these horrible names. Let me just go back a little bit, because they're named after the scientists who found them. So let me just go back one little bit to here. So what's happening is this atrium fills up with blood, and the contraction will spread out from the SA node, the sinoatrial node. It spreads out to the AV node. It can't spread out over the ventricles because they need to contract from the bottom. So it then goes down this specialized fiber called the bundle of His. His is just the name of the scientist who found them, and reaches these special fibers at the bottom, which go further into the muscle here, and they're called Purkinje fibers, and that's where the signal is sent up to those Purkinje fibers. And you can see then that ventricles contract from the bottom. from the bottom upwards. So that middle bit of the heart there, that those bundle of hairs, fibres go down, is called the septum. So it runs through the middle. And then we get the squeeze of the ventricles from the bottom. And that ensures that the blood then goes out through the aorta and the pulmonary vein. Now, each of these segments on this graph show the various parts of that contraction. So, for example, this is the atrial contraction in the P wave, you can see there. And then, so if you just look now, it will show you the atria fill in and contract in the P wave. Then the next thing to happen is the ventricles fill. Once they've filled, the signal can move down from the sinoatrial node, down the bundle of hairs to the Purkinje fibres, and then this big peak, the R wave, sometimes it's called the QRS section, is when we get the big contraction from the bottom of the heart, and that forces the blood upwards out of that bottom chamber, because the vessels that the blood needs to travel down are at the top of the ventricles. So we need the blood to squeeze from the bottom up, and that happens during this QRS phase. And then it goes off into in that ST section, it goes off into the aorta. You can see that now going as the heart contracts. Okay. So this sinoatrial node then is this is the specialized cells that are in the right atrium, and it has special properties that can begin this contraction, this wave of depolarization across the atria. And the word that is used to describe the fact that it's specialized and it can depolarize and it can initiate heart contraction is myogenic. So the heart is said to be myogenic. This tissue is said to be myogenic. And if you're asked a question that says, what does it mean for the heart to be myogenic? It means that it can initiate its own contraction. It will contract and relax spontaneously on its own. Other types of muscle, such as skeletal muscle, aren't myogenic. They don't contract from within themselves. They're neurogenic. They will only contract because they receive a nerve impulse via something called a neuromuscular junction. So it's a connection between a nerve and a muscle. So they aren't myogenic. Only the heart is myogenic. Cardiac muscle is very specialized, obviously, because it's got a very specialized role. That's the diagram of it there that you've been given. It's not great. You can see bands. It's not as striated as skeletal muscle, but you can see bands. There's lots of interconnecting cells whose surface membranes are joined very tightly together. So let's have a look then at these waves of electrical excitation that pass between these parts of the heart. So the individual muscle cells cannot be allowed to contract on their own natural rhythms, as we said before. We can't have one side doing its own thing. We can't have the atria and ventricles out of sync. The heart wouldn't work properly if that was the case. So it has to be all coordinated. And this is one of the very special roles of the SA node, these specialized cells, is they act as a pacemaker. So along with the other cells to which they're connected, the fibres, that SA node regulates the beat of the heart. So the muscle cells of the SA node set the rhythm for all of the other cardiac muscles. And each time they contract, they set up a wave of electrical activity, which will spread out over the walls of the atria to start with, then will spread down to the AV node, making sure that the ventricles don't contract just from touching the atria. So the atria contracts to get the slightly to get the blood into the ventricles, but we don't want the ventricles to contract from the top down. We need them to contract from the bottom up. There's a brief delay, and this conduction from the atrial cells to the AV node, the atrial ventricular node, you can see it there, look. So it starts off here in the sinoatrial node, spreads over the atrial tissue, the muscle tissue, the cardiac muscle tissue, reaches the AV node, the atrial ventricular node. And there's a brief delay while that blood can empty into the ventricle and fill, while the signal goes from the AV node down the bundle of his. And this bundle of his branches, there's two separate branches, one going around the right ventricle and one going around the left ventricle. You can see it sort of clearly in this image here, the branching. And these branches have got loads of little fibres that come off. It's not just this line. There are little tiny fibres that aren't really shown on this diagram that go in to the muscle as well, sort of like fingers almost. And they're called Purkinje fibres. There you go. You can sort of see it better there. Okay, on this diagram. So if I just go back and we've got the heart there, that's sort of where it would be. So the SA node is up the top in the right atrium. That will feed across the cardiac muscle of the atria to the AV node, the atrial ventricular node. So that's crossing between the atrium and the ventricles. And then the signal goes down the bundle of his to the Purkinje fibres, causing the contraction of these muscles from the bottom up, squeezing the blood upwards so that it leaves the heart via those arteries that are, if you have a look at this diagram, you know, they're pretty high up on the ventricles. Okay. Right, so I'll just play this for you. Just to have another look from another angle. So those are our atria. That's the SA node, obviously. And you can see the connections between it there. Really quick contraction of the top chambers there. And then the signal passes down that middle septum and initiates the contraction from the bottom. So the AV node is picking up the signal from the sinoatrial node, travels down this fibre, the bundle of his, which branches into two. Then the depolarization moves into the Purkinje fibres, which initiate the contraction of the ventricles there. So those big white things that you can see on this diagram are the valves that prevent flow of the blood. So here's our ECG, electrocardiogram. Sometimes it's called EKG. Not sure why they use a K, but sometimes they do. What it shows, as we saw in the video earlier, was the waves of excitation traveling across the heart, placing electrodes on the body and measuring the electrical potentials. So I'm not in slideshow. Right, there we go. Yeah. Oh, that's cool. So you saw this happening, did you? Or so essentially what you end up with is a graph, and you've got the electrical potential on the vertical axis, and you've got time on the horizontal axis. And what we can see is, starting from left to right, the P wave. And as we said earlier, that's the atrial depolarization. So it's the wave of depolarization or excitation, if you prefer, sweeping over the walls of the atria. And that's quite small compared to the next bit, the QRS complex, which is the depolarization and excitation of the ventricles. That's the big push, basically, of the blood out of the heart. The T wave is a tiny little lump after the QRS complex, and it's where the ventricle walls recover back to their original state. So you can see there. Continual heart beating. We need that, obviously, because we need to continually get the oxygen to all of our cells for cellular respiration. We can't survive without it. So if we hook someone up to an ECG machine, you get one of these readouts. You can see there P wave, QRS complex, and then the little T wave at the end. So it's really fast. It's really hard to look at the heart and the graph at the same time. But you can see the initial excitation there of the SA node is lit up during that sort of rest phase. And then during the P wave, it contracts slightly to empty, and the excitation is moving over the tissue. So it's atrial systole, so the atria is filling, but we're getting excitation. Then we've got a little rest phase where we're not seeing excitation in that PR segment. And then the QRS bit happens really fast. The AV node has picked up the depolarization, that picked up the signal, travels quickly down that bundle of hairs down into the Purkinje fibres. That's that big QRS section of depolarization. Contraction of the ventricles. And then we are in ventricle systole, so that's the squeeze during that time. And we've got a rest where there's no depolarization happening for a bit. And then we've got the last little bit of T phase where everything's going back to fill up in ventricular diastole, ready for the next squeeze. So what have we done so far? So we've had a look at the need for a double circulatory system and the demands of the human body. We've had a look at the arteries and veins the last session and had a look at fetal blood the session before, had a look at the components of blood as a liquid. That was a couple of sessions ago. What I'll do now, because we're at the end of that, I will, if you've got any questions, you can ask them, obviously, but I will get up the next slides so we can make a start on that as well. Right. So are there any questions about those contractions and excitation and moving the signals across the heart or anything before I move on to this bit? Okay. So cardiovascular system then is basically focusing, as we've seen, getting glucose and oxygen around the body. So we're going to have a look at the actual oxygen exchange next. We've talked a lot about getting the blood pumped to the lungs. Obviously, that's for picking up oxygen. And we're going to have a look at what happens and how that happens next. So we haven't got a huge amount of time, so I'll just sort of introduce the first bit. First of all, we need to clarify the difference between breathing and respiration. It's not great because we call this the respiratory system, but respiration isn't breathing. Breathing is how some animals get the oxygen they need for a cellular process called cellular respiration. So we breathe to get the reactants we need for cellular respiration. And cellular respiration, or just respiration, is how we get the energy from glucose. It's a reaction. Well, it's not a reaction. It's a series of loads of reactions where we release ATP, which is a molecule, but it's a really high energy molecule. It's a reaction, how we release this really high energy molecule from glucose. And oxygen is usually needed. You can do it without oxygen, but it's a bit rubbish. It's nowhere near as fast or efficient to do it without oxygen. So we can do it without oxygen because sometimes we can use up all the oxygen in our blood, like if we work really fast, really hard for quite a long time. And we get lactic acid in our muscles if we do that. So if you've been running really fast and hard at the end of a long distance race, for example, you might have, you know, if you've got to do a sprint at the end of the 1500 meters in PE, imagine, you might have used up all the oxygen that is currently in your blood, but you're not going to die from doing PE. What's going to happen is your body is going to switch to a type of respiration where your cells can keep working and making this ATP energy, but without oxygen, but for a really short period of time. And then you collapse on the floor and you breathe really deeply. You've got lactic acid in your muscles. They're going to hurt, and you can recover that oxygen by breathing deeply and getting some more back into your system. So cellular respiration is releasing ATP energy from glucose. You usually need oxygen, but not always. So we're going to have a look at the respiratory system and how we can get that oxygen into our cells for those cellular respiration reactions. So first of all, we have to get the air into our lungs, and we do that by breathing it in. And the first place that the air comes into is our nostrils. And in our nostrils, the air is filtered to remove foreign particles. You'd be surprised how much foreign particles you breathe in. So pollen, for example, dust is another example. If you've ever gone anywhere really dusty, you'll find it in your mucus afterwards. It's not very pleasant, but it all gets trapped in your nose. So The nose is really important for filtering out all of the stuff that we accidentally breathe in. There's an awful lot of dust and dirt and pollen and quite often bacteria and viruses as well. And not only does our nose act as a filter, actually it's not just specialist cells and mucus that we've got there, but there's hairs in our nose as well. They're really important to help filter. But not only does it act as a filter, but it warms and moistens the air that comes in as well. And we've met some of these specialized cells before when we were talking about epithelial cells with specialist structures on them. Let me just check the chat. There's a large conversation around microplastics being inhaled this day and age as well. Yeah, I'd imagine that they are, to be quite honest with you. I can't imagine that they're not. They're everywhere. They're in our water, they're in our food, they're in the air, they're in our blood, they're even being found in fetuses. So yeah, that I imagine we are breathing plenty of microplastics in. So first structures are the nose. There is lots of adaptations to help us deal with what's being inhaled. For example, we've got a mucus layer and we've got ciliated cells that have tight junctions between them. And what happens is all of these particles get trapped in the cilia. They act as a wave, get trapped in a mucus, sorry, in the cilia. They act as a wave and move the mucus over the cells. It's a bit disgusting. They move it to the back of your throat where you swallow it and it ends up in your stomach. And if you think back to the digestive system sessions, what's in the stomach? Loads of acid, really low pH, horrible environment for bacteria, very acidic, hopefully killing off most of the horrible stuff that we've breathed in and then subsequently swallowed. So the outside air is entering the respiratory system. It's moistened, it's warmed, and all these horrible bits of dirt, pollen, probably microplastics, and everything else gets swallowed into our digestive system. So the particles have to be bigger than 5 micrometers to be trapped by hairs in the nose, and then the others are caught in the mucus. And these are produced by special cells called goblet cells. Remember, we had a look at goblet cells when we were talking about epithelial tissue, a special type of specialized epithelial tissue right at the beginning in the first section. And these ciliated cells have tiny, they say hair-like projections. They're not hairs at all. They just look like hairs. They're more like just sticky out bits of the cell. And they continually beat to move this mucus layer along. They create sort of waves where it's all pushed along and then swallowed down the esophagus, where everything's broken up by this really strong enzymes in the stomach and that acid as well. So I think that would be a good place to leave it on there. If you've got any questions, let me know. If not, we will pick this up next session. Okay, bye guys. Bye-bye.